Hey everyone, welcome to Introduction to Cognitive Science. I'm Adam, and the focus of our lecture today is on introductory neuroscience and neuroscience methods. One of our main goals for today is to learn some basics about the brain and its functions, as well as methods for studying the brain and its functions. A few methods that we'll look at today include lesion studies, structural magnetic resonance imaging, functional magnetic resonance imaging, EEG or electroencephalography, and TMS, transcranial magnetic stimulation. I'll start off by talking about lesion studies. Okay, so we have a question. How can we study the brain and its functions? How do we know what parts of the brain are responsible for what cognitive functions? Well, one way that we can approach that question, one way that we can find an answer to that question is by studying individuals with brain damage or lesions. So for example, we might find military veterans or others that have suffered brain damage through accidents, and we can associate their brain damage with an impairment in their cognitive functioning. So for example, imagine that through some accident, I received damage to my amygdala. Okay, maybe I'm a construction worker and some sort of metal part, hit me in the head and damaged my amygdala. Well, in this case, since we know that the amygdala is damaged, we can then look at behaviors that are impaired as well. Okay, and by studying people with amygdala damage, we might find that in general, they also experience or demonstrate impairments with emotional regulation. Okay, and then we can associate the damaged or lesioned amygdala with impaired emotional regulation. Okay. Similarly, we might find other individuals that have damaged hippocampus or damaged cerebellum, or pick your favorite part of the brain and imagine that it's damaged. Then what we do is we just find through systematic investigations which emotional or cognitive aspects of their behavior are also impaired. Okay, and by associating cognitive mechanisms or areas that have been lesioned and the functions that are impaired, right, the associated functions that are missing in those individuals, we can start to have a better understanding of each cognitive component or each component and its associated function. Okay, and that's how we can, through lesion studies, get an idea of brain components and their associated functions. Okay, a strength of this approach is that it gives us a good sense of causality, right? If you're missing an amygdala and you are missing emotions, and other people have an amygdala and have emotions, that's pretty compelling evidence that the amygdala plays an important role in emotional processing. Another strength of this approach is that it makes good use of individuals that are already suffering these impairments, right? So maybe that there's a veteran who had damage to a part of their brain. Well, by offering themselves to scientific investigation, we can get a better understanding of what's impaired, and perhaps we can investigate how to repair that brain component and recoup some of those cognitive functions. Okay. So I think that that's nice that we can always sort of look at, even in our unfortunate cases, right? We can still be productive and useful in mining those for information and trying to generate some useful solutions for the future. Okay. Of course, some drawbacks or limitations of this approach is that we can't induce lesions in participants, right? That would be highly unethical to do so, right? I can't, for example, get 100 of my students from Berkeley and induce lesions in 50 of them, and then have another 50 without lesions, and then have them all perform a cognitive task and then see who performs better on the task, right? That would be highly unethical to put my students through such a situation. Okay, so we see limitations clearly in the lesion approach. All right, we have other approaches in addition to the lesion approach. We have structural magnetic resonance imaging, or MRI. And this is a method that's used to take pictures of the brain's anatomical structure. Okay, so as you can see in the bottom right figure on the slide, we have some static pictures of the brain, right? Different slices of the brain. MRI is nice because we can get a nice static structure or picture of what the brain looks like. Okay. This is importantly distinct from fMRI, or functional magnetic resonance imaging, and that fMRI is going to not look at the brain as it is statically, structurally, but it's going to look at the brain as it's engaged in a cognitive task, right? As it's functioning in a cognitive task. Okay, that's why we call it fMRI. Okay. The way that fMRI works is that it's going to measure blood flow or the bold response in the brain as your brain is engaged in a particular cognitive task. Okay. The bold response is the blood oxygen level dependent response. Okay, this is a response that's based on your blood flow and blood oxygen level, right? The way that I like to explain this, how the bold response works, or how fMRI is used to measure blood flow and the bold response, is consider you're doing push-ups, right? Well, just at rest before you do the push-ups, you have a certain amount of blood flowing through your body, right? Just imagine that it's like an equal distribution, okay? Now, I wanna give you a certain task to perform, okay? Say that task is push-ups, okay? Drop down and give me 20 push-ups, right? As you drop down and you are engaged in that physical task, notice that you're gonna get increased blood flow to the relevant sites, right? Your pecs, delts, and triceps, for example. Okay, those are gonna be your major movers when you're doing push-ups, okay? As you engage in your push-ups, you're gonna get increased blood flow to those sites. And in addition to that blood flow, you're gonna get increased blood oxygen level dependent response, okay, or a bold response, okay? And we could use imaging techniques to measure that blood flow. And we would see that, aha, when you're doing push-ups, indeed, we get increased blood flow and bold response to the pecs, shoulders, and the triceps, okay? And that would let us know that when we're engaged in that task, these are the muscles that are active, or these are the regions in our body that are active, okay? On the other hand, if I was doing pull-ups or rows instead of push-ups, we would get activation in very different areas, right? We would get activation in the latissimus dorsi as well as in the biceps, right? And we could use imaging techniques, right? Techniques that measure blood flow and the bold response to verify that that's the case, right? In addition to us just having a nice pump in those areas from doing push-ups and pull-ups respectively, okay? So that's the general idea behind using functional magnetic resonance imaging, right? While we're engaged in a function like push-ups or pull-ups, or like a lexical decision-making task, or some other task, right? Cognitive task, we can look and see what areas of the body or the brain in the cognitive case is selectively activated for that cognitive task, okay? And a nice thing about fMRI, it has excellent spatial resolution, and it provides us with some really beautiful images of the brain as it's engaged in some function, okay? Some limitations of fMRI is that compared to electroencephalography or EEG, it's rather slow. It's a little slow compared to EEG, okay? So in terms of its temporal resolution, it's a weaker method than EEG, which is going to have finer temporal resolution. Okay. So this segues nicely into our next methodology, which is electroencephalography, or EEG. And this is a method that's used to measure electrical activity produced by neurons in the brain during different cognitive tasks. So just like when you're engaged in a cognitive task, maybe it's a linguistic task or an executive control task, okay? Just, pick your favorite cognitive task, and we want to figure out what area of the brain it engages, okay? Whereas fMRI would look for blood flow and a bold response while engaging in that task, EEG is going to look for electrical activity, okay, from the neurons, okay? And neurons are nerve cells, which we'll discuss here in subsequent slides, okay? One of the nice things about EEG, right, and we saw this as we discussed the relative weakness or limitation of fMRI, is that EEG is quick. It has excellent temporal resolution. So if you're investigating something where maybe spatial resolution isn't so important in this task, but temporal resolution is super important, okay, then EEG is going to be an excellent way to go, all right? Unlike blood flow, right? If I tell you to drop down and give me 20 push-ups, when you drop down and you start pushing, it's going to take a moment for the blood to start flowing, right? For that bold response to be picked up through fMRI. On the other hand, electrical activity, right? When you turn on the switch to turn on the light in your house, notice how quickly that happens, right? Likewise, neural activity, right, will be picked up via EEG in a much quicker manner than the bold response will be picked up through fMRI. Okay, another way to say that is that temporal resolution is excellent for EEG, but a relative trade-off is that spatial resolution is not so great compared to fMRI. Okay, so fMRI and EEG, they have the opposite strengths and weaknesses. Okay, so in many studies, when it's possible, we'll see researchers trying to utilize both, or maybe they utilize one approach in the first study and the other approach in the subsequent study, right? Trying to narrow in both spatially and temporally, right, on what's doing what when, okay, in terms of brain and cognitive function. Okay. All right, and we'll see some pictures of fMRI and EEG, the sort of images that they provide us with here in the next slide. Okay. And the last method that we're going to talk about for today is transcranial magnetic stimulation. Okay. And this is a method used to apply magnetic pulses to stimulate neurons or nerve cells in areas of the brain. So whereas fMRI and EEG, they're going to get you to perform some task first, right? I'll have you do push-ups, right? Or a lexical decision or executive control tasks, right? And then I'll see what area of the brain is active, selectively activated for that task. In TMS, I'm going to go ahead and stimulate an area of your brain, okay? For example, I might stimulate Broca's area in your brain, right? And we might see that has a facilitative effect on linguistic processing. And here, what we're doing is we're stimulating, right? We're using magnetic pulses to stimulate the brain and then see how that stimulation results in some difference in function, right? So if I apply TMS to one group and another group does not have TMS applied, we might see that the group with TMS has improved function, whereas those without TMS have normal function, right? The group without TMS would be our control group, and then the group that is receiving TMS would be our experimental group, right? And then we could see maybe by stimulating certain areas of the brain, right, we might see that that facilitates or has some impact on cognitive function. Okay, we've seen actually TMS has been shown to help with mood disorders and other. It's a nice form of treatment for different things. Yeah, so we can see on the top right is a picture of TMS and how that works. Okay. So we see that we have, oh, before I move on, notice that TMS, transcranial magnetic stimulation, is somewhat like a lesion study, right? In that way, we're turning something on or turning something off with the TMS, right? Whereas with the lesion, it's permanent, right? We're removing something for good, okay? With TMS, this is only a momentary phenomenon, okay? What's interesting is that with lesion studies, because the brain has an amazing ability for neuroplasticity, what we find is that oftentimes a person that has like brain damage and has certain cognitive functions disrupted will over time be able to recoup some of those functions, right? Because other parts of the brain will start to help support and aid in that functionality. Okay, in TMS studies, we may be able to turn off or disrupt a cognitive function, and we wouldn't know just from that study using TMS what sort of the more long-term change might be, right? Like we might show that, aha, using TMS, we can disrupt this area and that turns off this cognitive function. Right. And so we might infer that, if we gave a lesion there, it would also disrupt that function. My point here is that because lesions are long-term, whereas TMS is only short-term, the nature of lesions allow for some plasticity to occur, right? Given certain qualifications, okay? So I think that that's really interesting to appreciate these different methods, when to use different methods, what the strengths and limitations are for each method, right? This will help us decide what methods are best given our particular situation, what we're trying to show in a particular cognitive science experiment, okay? All right, let's move on now to our next slide. And here on this slide, we have a nice visual of the difference between functional magnetic resonance imaging, which is on the left, and electroencephalography, which is on the right, okay? And here we see that this is tracking activity in the brain while participants are engaged in a linguistic task, a music task, or a motor imagery task, okay? And notice that with fMRI, we get some beautiful images of the brain, right? Look how detailed those images are, right? That's really nice. On EEG, you can't see so much of that detail, right? That spatial resolution is lacking in the EEG. However, we'll see that the temporal resolution in EEG is excellent, right? And this is made clear in this table right here. So in this table, we see that I just want to point out fMRI and EEG for today. And of course, in future lectures, we can always focus on other approaches. But it's nice to be familiar with fMRI and EEG. So I want us just to appreciate the different strengths and weaknesses of each approach. So I've provided the spatial and temporal resolution for both fMRI and EEG. We see that the spatial resolution of fMRI is 1 to 5 millimeters, but the spatial resolution of EEG is 20 to 30 millimeters. So we see that the spatial resolution of fMRI is much finer than the spatial resolution of EEG, right? 1 to 5 millimeters for fMRI and 20 to 30 millimeters for EEG. Okay. However, look at the temporal resolution. So the temporal resolution of fMRI is 1 second, whereas the temporal resolution of EEG is 1 to 4 milliseconds. So we see that in terms of temporal resolution, EEG is superior to fMRI. So I just put here as a summary, fMRI has better spatial resolution, but worse temporal resolution than the EEG. And the EEG has better temporal resolution, but worse spatial resolution than fMRI. So depending on the nature or purpose of your study, fMRI or EEG may be a better option, given the relative strengths and weaknesses of each methodology. All right, next, now that we've looked at some methods for studying the brain, next let's go ahead and discuss some basics of neuroscience. For example, what's a neuron? That might be important for our work as cognitive neuroscientists. So neurons are nerve cells that are fundamental units of the brain and nervous system. The soma or cell body of a neuron includes the nucleus. So we can see in this figure, we have the soma here, and that includes the nucleus. And the nucleus contains genetic material of the cell, right? And we see that that genetic material is the chromosomes. We'll also notice here that we have dendrites and an axon. Okay, so the dendrites are here and the axon is here. Dendrites receive information from other neurons, right, and convey it to the soma. The axon carries information away from the soma and sends it to other neurons. Okay, so it's going to transfer information from the soma to the end of the neuron and then to other neurons from there. Also, we have the myelin sheath, which is a layer of fatty tissue that covers the axon and helps to speed up the transmission of information. Although all neurons have the same fundamental structure, they play distinctly different roles in terms of the type of information they transmit. Sensory neurons transmit information from sensory cells or receptors to the brain. Motor neurons transmit commands from the brain to the glands or muscles. Interneurons connect neurons to each other. And mirror neurons respond similarly when we perform an action and when we observe someone else performing that same action. Okay, so when I punch, like if I'm boxing and I punch, or when I see someone else punch, right? A mirror neuron is the type of neuron that's active both in the action and observation. Okay, and those are really cool. So neurons rely both on electrical impulses and chemical messages to convey information. And we see here, we have a figure of a chemical synapse and an electrical synapse. The action potential is a change in membrane potential that travels down the axon in an all or nothing manner. When a neuron fires, it releases neurotransmitters, which are chemicals that are capable of attaching to particular receptor sites, of receiving neurons and activating them. And we know that we have different neurotransmitters. I'll discuss some different ones here in a few slides. But also, if you take a course on addiction or on drugs, you'll learn a lot about different neurotransmitters. And this is a very interesting topic. An agonist is a substance that binds to a receptor and triggers a response that mimics or enhances a neurotransmitter's effect. This is like an on sort of switch, right? An antagonist is a substance that binds to a receptor and triggers a response that blocks a neurotransmitter's effect. It's like an off. So that's the difference between agonists and antagonists. So the nervous system uses neurons or nerve cells and neurotransmitters to send messages through the body. The nervous system, we can think of it both in terms of the CNS and PNS, or the central nervous system and the peripheral nervous system. The central nervous system includes the brain and the spinal cord, hence its title as central nervous system, right? This brain right here, and sort of the little tail of the brain, right? The spinal cord. And we see that in this figure, it's highlighted right here in this sort of lighter tan color. Okay, the PNS, or the peripheral nervous system, includes all the nerves and ganglia located outside of the brain and the spinal cord, or in other words, outside of the CNS. So all the blue wiring right here, right? If we want to build a person, right? The blue wiring is the peripheral nervous system. In addition to the nervous system, which uses neurons and neurotransmitters, we also have an endocrine system, which uses glands and hormones to send messages through the body. The pituitary gland in the brain is usually considered the primary endocrine gland because it releases hormones that activate the other endocrine glands. Other glands include the thyroid in the neck, thymus in the chest, adrenal glands and pancreas near the kidney and stomach, and the gonads, ovaries and testes. And we see the endocrine system over here in this bottom figure. So we'll see that the body, the human body, is an amazingly integrated and complex system, right? With, different aspects of the nervous system. We have a nervous and an endocrine system. We use electrical information as well as chemical information, right? We're going to have faster components and slower components. And we see that this is a marvelous system, a lot of parallel and distributed processing going on in the system, okay? Very different than like a computer, right? A sort of a computer that executes in linear fashion, okay? So let's go ahead and look at some of the neurotransmitters and hormones. And we'll just get a basic idea of what they are involved in. So some neurotransmitters include acetylcholine, and this is a neurotransmitter that's involved in arousal, attention, and movement. Dopamine, this is an important one for us cognitive scientists of language, because dopamine is involved in anticipation of pleasure, but also fine motor control and sequential thought, right? We also have GABA, or gamma aminobutyric acid. You just call it GABA. And this is an inhibitory neurotransmitter of the central nervous system, particularly of interneurons. All right, so GABA is going to be one of the major inhibitory neurotransmitters. Glutamate is an excitatory neurotransmitter with critical roles in learning and memory. Okay, so it's important to understand that GABA and glutamate are two important neurotransmitters. One is excitatory, glutamate, and one is inhibitory, gamma aminobutyric acid, or GABA. And another neurotransmitter is serotonin. This is a neurotransmitter that's involved in aggression, depression, impulsivity, and sleep onset. Some examples of hormones include the following. Cortisol. This is a hormone that's involved in managing blood sugar, stress, and inflammation. Having a lot of cortisol can be bad for you. Epinephrine or adrenaline is a hormone involved in physiological arousal and energy. You might have heard stories where people had a lot of adrenaline or a shot of epinephrine and they were able to like lift the front end of a car to save a baby or some other amazing physical feats. Estrogen is a hormone involved in regulating emotional well-being and the reproductive system in women. Oxytocin is a hormone involved in social bonding, friendship, and love. And testosterone is a hormone involved in the creation of new blood cells and the regulation of the reproductive system in men. All right, next, let's go ahead and look at the brain and how it's divided into major sections. So the brain can be divided into three major sections: the hindbrain, the midbrain, and the forebrain. The hindbrain is the part of the brain directly above the spinal cord. So you can see in this figure, we have the spinal cord right here, and then boom, the hindbrain is sitting right on top of it. Okay. The hindbrain directs basic processes needed for survival, such as respiration, heart rate, and arousal. The hindbrain consists of three separate parts: the cerebellum, medulla, and pons. The cerebellum, which we see back here, controls fine muscle movement and balance. The medulla is essentially an extension of the spinal cord and conveys messages to and from the brain. And the pons carries information between several areas of the brain. Next, we have the midbrain, which is located above the hindbrain. Okay, so we're making our way from the spinal cord up. Okay, and then the forebrain will be the last. Okay. Running through the hindbrain and midbrain is a network of neurons called the reticular formation. And this is a very cool area of the brain because it maintains consciousness and regulates behavioral arousal and alerts other parts of the brain to important events. The midbrain also contains a small structure known as the substantia nigra that secretes the neurotransmitter dopamine. Okay, and we already discussed how we love that. So the midbrain, there's some things to love about the midbrain: the seat of consciousness and an important area for the secretion of dopamine. And the third largest part of the brain is the forebrain, which controls thoughts, motivations, and emotions. The forebrain is involved in complex emotional, cognitive, and behavioral processes. The forebrain includes four key structures: the limbic system, the thalamus, the hypothalamus, and the cerebral cortex. And we'll talk about each one in turn. The limbic system includes the amygdala and hippocampus and is involved in emotions and memories. Okay, so we can see in this figure here, the amygdala and the hippocampus. Amygdala is largely involved in emotions, especially negative emotions, and the hippocampus is an important site for memory. Next, we have the thalamus, which is involved in receiving information from various sensory systems throughout the body and directing this information to the appropriate parts of the brain. We see the thalamus is right here. The hypothalamus is involved in controlling temperature and many basic motives and drives, including hunger, thirst, sex, and aggression. And the hypothalamus is right here. And then finally, we have the cerebral cortex, which is involved in controlling more complex behaviors and higher mental processes. And we'll go through the lobes and whatnot here shortly. All right. So we see the lobes are provided here in this figure, but also in this figure right here. And I provided this figure in addition to this previous one, because unlike this one, on this figure, we have some nice points of separation. I'm gonna pause the video real quick while the gardener moves by. All right, so on this figure, we have the 4 lobes of the brain, as well as some nice divisions. Okay, so we see that there's the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. Okay, the occipital lobe, the lobe in the back right here, is, so what I want you to do today is sort of just get like a big picture idea of important things that are going on in each of the lobes, right? We'll see when we get to Brodmann's area that there's lots of more distinct areas, right? It's not just sort of 4 parts of the brain and that's it, but we have finer levels of analysis, okay? So I don't want you to understand, the goal of today is not to memorize all the Brodmann areas, right? But rather I want you to sort of understand the 4 lobes of the brain and then try to memorize a particular important function in each of those, okay? And start there. And after you feel very confident, okay, I can divide the brain into the major lobes and identify salient functions for each of those, then go ahead and add to that, okay? Like, oh, okay, well, in addition to temporal lobe being important for auditory processing, there's some other things going on there too, okay? But for today, I want you just to tie at least one function to each of the lobes. So the occipital lobe is important for vision, okay? So visual cortex is here. The temporal lobe on the sides right here, important for audition, right? This is the home of auditory cortex, okay? I have a fat head, so of course I love music, right? The parietal lobe, as we see right here, is important for somatosensory information, right? We'll see that there's a map, right? A somatotopic map of like the hand, foot, mouth, and whatnot in this area, okay? And then the frontal lobe right here is important for motor actions, okay? And we see that the motor cortex is here. In addition to these major lobes, we can use the fissure and sulci to distinguish the lobes of the brain, okay? So the central sulcus, often called the central sulcus of Rolando, this separates the frontal and the parietal lobes, okay? And we can see right here, this line is partitioning these 2 lobes, okay? The parietal occipital sulcus right here separates the parietal from the occipital lobe, okay? And the Sylvian fissure right here separates the temporal lobe from the frontal and parietal lobes. All right, that's how we can nicely separate these major lobes of the brain. All right, and here we see that whereas in the last figure, we had a division of the brain in terms of lobes, here we now have the division of the brain in terms of Brodmann areas. Okay, and I think that this is really cool. Each of these Brodmann areas are labeled BA and then a number. Okay, so we'll see that although it's correct to identify the occipital lobe as the home of visual cortex, we can also identify it with BA17. Okay, so this area of the brain right here. Okay, and this will help us identify like primary visual cortex from secondary and association areas. Okay, and although in the last slide we saw that the temporal lobes are the home of primary auditory cortex, we also see that BA41. And we can also identify that area right here with BA41. okay, auditory cortex. BA3 is somatosensory cortex, and then BA4 next door, but a little bit more forward, right? Going into the frontal lobe, we have motor cortex, okay? All right, and as a fun exercise, I encourage you to try to identify all the Brodmann areas, right? What's 10, what's 38, what's 19, and so on, okay? Some things that you can do is ask yourself, for example, BA10, what lobe is that located in, right? Is it in frontal lobe? Is it in occipital lobe, right? So you can, at a more coarse grain level, just make sure that you know where the BAs are in terms of lobes, but also you can then, in a more fine grain manner, ask, what is this BA region responsible or at least partially implicated in, right? Is this area involved in audition, or is it involved in vision, or so on, okay? All right, cool. And we'll talk a lot about this, especially tomorrow when we get into the cognitive neuroscience of language. All right, so we've been talking a bit about brain components and brain functions. Here, I want to step back and also point out that sort of like the contralateral nature of brain and behavior. I think this is really cool and fascinating. So the left hemisphere of the forebrain controls the right side of the body, and the right hemisphere of the forebrain controls the left side of the body. So the left and right hemispheres of the brain control opposite sides of the body and are connected via the corpus callosum, this fiber tract here in the middle, okay? And so this is really cool. Like you can see here in this figure that this is the left side of the brain and the left hand, and this is the right side of the brain and the right hand. But notice that you see the left hand is being processed in the right side of the brain, and the right hand is processed on the left side of the brain. Okay. Also, we'll cover this when we get to vision, right? But we also see that the left visual field is processed in the right hemisphere, right? And the right visual field is processed on the left hemisphere, right? And you can, this is color coded. So, right, we see that the blue over here, right visual field, yeah, is this side of the visual field on the right side, okay? And the opposite for over here, left visual field is being processed over here, and the left visual field is on this side, okay? So I think that that's really neat. And pointing out the importance of the corpus callosum as this highway or information strip connecting both the left and right hemisphere. All right, now that we looked at some neuroscience methods, we talked a bit about neurons, the nervous system and the endocrine system. We talked about the different regions of the brain and some of their important functions. What we're going to do for the rest of the lecture is we're going to talk a little bit about vision and audition in particular, okay? And if this was a neuroscience course, of course, we would spend much more time in each area, right? We would spend many more lectures on vision and on audition in particular. But because this is an introduction to cognitive science course, we're just trying to grasp enough of the material, enough of the fundamentals that way we can understand higher cognitive functions, okay? Always remember, when we're doing cognitive science, what's essential, important to us is cognitive function, okay? It's very easy to get really into, for example, lower level visual processing, okay? But as cognitive scientists, we want to get what's relevant from that for some higher level cognitive process, okay? And not just sort of like studying cones for cone's sake, right? If you're interested in rods and cones for their own sake, then that's more of like the neurosciences, okay? All right, so we're gonna focus on vision and audition today, so that way when we talk about the cognitive neuroscience of language tomorrow, we can discuss language with some rudimentary knowledge of vision and audition. Also notice that what's really cool about Intro to Cognitive Science is that we can literally stop and zoom in any section of the course, right? Like we covered sign language briefly, but we could have zoomed in and spent a whole course on sign language, right? We spent a little bit of time on syntax, but we could have zoomed in and spent a whole course on syntax, right? Similarly, when it comes to neuroscience, we're just covering enough to get a basic understanding, but we can always zoom in and gain richer insights here. Okay, I encourage you to do so. All right, so in visual perception, a visual image is first projected onto a flattened sheet of photoreceptor cells that lie on the inner surface of the eye, the retina. It's cool to remember this because vision is already three-dimensional for us, right? But it's amazing to realize that the image is first projected onto a flattened sheet, right, of photoreceptor cells, right? And we, in some sense, construct vision from that, which I think is really amazing. So binocular fusion is the process of producing a single image from the two monocular images, right? And again, this is super fascinating that our experience of the world is integrated and unified and not broken in piecemeal like it maybe should be, right? Given that we have a blind spot, right? We have areas of vision that we can't see, right? Like a lot of our information, if you're noticing across the different studies, we see that a lot of our experience of the world is impoverished, right? Our linguistic experience is impoverished. Our visual information in some sense is, right? But we are able to systematize, right? And form a coherent experience of the world, which is really interesting and amazing. And binocular fusion is just this process of forming a single image from two monocular images. The central visual field, right? The visual field that's center is color sensitive, has high visual acuity, and operates at high levels of illumination or light. Okay, and we see here that this is our central visual field. Okay. The peripheral visual field is relatively color insensitive, not so sensitive to color, has poor visual acuity, and is more sensitive at lower levels of illumination. So one we'll see is great for daytime seeing, and another is great for nighttime. Okay. And visual acuity just refers to the ability to detect and recognize small objects. And color vision is the ability to detect differences in the wavelengths of light. All right, and here we just see sort of the monocular field from the left eye, and then the monocular field from the right eye, sort of like how much of the field of vision is accessible to each eye. And then with binocular vision in the middle, we have that nice overlap in the middle right here. Okay, and our visual acuity is going to be best in this center area. Okay, and also we're going to have great color perception. So the human visual system has two subsystems. And are we noticing subsystems within systems in the human body and the human cognitive system, right? This is really amazing, okay? The human visual system has two subsystems that operate at different levels of light energy. And over here, you'll see I provided a figure of rods and cones for you. Okay. So the scotopic visual system is a dark adapted system that operates at low levels of illumination. And rods are responsible for the initiation of the scotopic visual process. The photopic visual system is a light adapted system that operates at high levels of illumination. And cones are responsible for the initiation of the photopic visual process. Okay, so cones are for color. Okay. And rods are helpful for nighttime. Here we see that humans have a trichromatic visual system where visible colors can be created by a mixture of blue, green, and red lights. So here's a graph showing the absorbance spectra of rods and 3 types of cones. Excuse me. So here we see that on the x-axis, we have wavelengths of light in nanometers. And the y-axis, we have retinal response, percent maximum. Okay, so blue cones are maximally responsive at 419 nanometers. Rods are maximally responsive at 496 nanometers. Green cones are maximally responsive at 531 nanometers, and red cones are maximally responsive at 559 nanometers. All right, so we'll think about how these parts are integrated now or work together. So here are our rods and our cones, okay? And in the next few slides, we'll talk about the bipolar cells, the horizontal and amacrine cells, and the ganglion cells, okay? So in the visual system, there are at least 2 types of bipolar cells based on their responses to photoreceptor stimulation. On bipolar cells are hyperpolarized by glutamate, all right? So depolarized is like turning off, hyperpolarized is like turning it on, like super on, right? So on bipolar cells are hyperpolarized by glutamate, right? Remember that glutamate is excitatory, and it functions to detect light objects in a darker background. We also have off bipolar cells, which are depolarized by glutamate, and they function to detect dark objects in a lighter background. All right, so we just talked about rods and cones and then bipolar cells. Okay, now we'll talk about horizontal cells, which are this right here, okay? See the bipolar cell, and this is the horizontal cell. We're talking about this now. Okay. Horizontal cells have large receptive fields involving both a presynaptic contact with a small group of photoreceptors and a postsynaptic contact with a larger group of surrounding photoreceptor cells. Okay. And the presynaptic contact is axonal, and the postsynaptic contact is dendritic. By controlling the responses of the center photoreceptors based on the responses of the surrounding photoreceptors, the horizontal cells indirectly produce the bipolar cell receptive field surround effect. Okay, so the horizontal cells help produce a surround effect, which helps to enhance brightness contrasts and helps to produce a sharper image, right, by enhancing brightness contrast. Okay, so you can sort of think of this as like a feature on your phone that allows you to enhance contrast in the picture, right, to produce a sharper image. You could ask, what allows you to do that in your phone? It's the horizontal cells, right? If your phone was really your eye, right? In other words, what is it about your eye that allows you to sharpen the image by enhancing brightness contrast? Well, in our visual system, It's the horizontal cells, okay? So the surround effect produced by the horizontal cell is weaker than the center effect. The surround effect produced by the horizontal cells enhances brightness contrasts to produce sharper images, to make an object appear brighter or darker depending on the background, and to maintain contrasts under different levels of illumination. Amazing, right? All right, so we've talked about rods and cones. We've talked about bipolar cells and the horizontal cells. Right, now let's talk about amacrine cells and ganglion cells. So amacrine cells modulate the synaptic activity of the retinal bipolar and ganglion cells, thereby affecting the transmission of visual information by ganglion cells. Retinal ganglion cells possess voltage-gated sodium channels in their axonal membranes and generate action potentials when they are depolarized by the glutamate released by the bipolar cells. In most neurons, depolarization results in neurotransmitter release by the bipolar cell at its axon terminals. Most bipolar cells release glutamate, which is excitatory to most ganglion cells. And types of retinal ganglion cells include at least the following two, okay? Type M and type P. Type M retinal ganglion cells are color insensitive motion detectors, whereas type P retinal ganglion cells are color sensitive object detectors, right? So type P are great at detecting objects and are sensitive to color. Type M are not so sensitive to color, but pick up on motion, okay? Right, so you may not, like if there's something way on the peripheral, you may not be able to identify what color it is or like what object it is exactly, but you can see it move, right? Like, I noticed motion over here, okay? On the other hand, if it's right in front of you, you're gonna be able to be sensitive to the color and the type of object that it is. So very cool, right? We have different types of things working in a coordinated, systematic way. And one of the reasons I'm pointing out the complex, systematic, yet specialized nature of the brain, right? We notice, for example, that in the brain and in the body, there's lots of different components. They all serve different functions, right? Specialized for different things. Like, hey, I'm a rod, I do color, or I do, I'm a cone, I do color well, or I'm a rod, I do like motion well, right? We see that there's different things that are specialized, right? We may ask a very important question is, how does the brain and the body become equipped with such specialized equipment, right? And how does it integrate this equipment in such a useful, coordinated way, right? And it's important to ask that question because we can start thinking about answers to that next week when we cover evolutionary psychology, right? Evolutionary biology and evolutionary psychology, okay? Although when we're doing like cognitive neuroscience, we can take neuroscience as given and investigate what's going on with the mind. There's always room for like a rotation where we may have taken neuroscience for given, like not have questioned why are rods and cones specialized for what they're specialized for, right? When we're doing cognitive neuroscience, we just accept that they're specialized. What I'm trying to say here is that in cognitive science, we can always sort of do a rotation and then ask at a later time, why is neuroscience the way that it is, right? And we can approach that from an evolutionary perspective or via work in anthropology, okay, and biology, which is really cool, right? Because once again, tying this back to our very first lecture, we saw that one of the great things about cognitive science is that it's a multidisciplinary endeavor, right? And so whereas if you're only a philosopher or you're only a neuroscientist, it may be slightly easier to sort of be stuck in a single frame of mind, right? But as cognitive scientists, we can always do an oscillation and self-interrogate, right, some of our previously assumed positions. Okay. All right. So I think it's very cool that we have these different types of rods and cones, horizontal bipolar amacrine ganglion cells, and even among the ganglion cells, we see that there's different types. Okay. All right. So we touched upon this briefly, but V1 or visual cortex, we know that's in occipital lobe. This is also called a striate cortex. All V1 neurons respond to visual stimuli exclusively. Okay? That's why we call them V1. This is primary visual area. Electrical stimulation of V1 elicits visual sensations, right? So if you stimulate that area, you might see some cool things. And then ablating V1, right? If you cut it out, this is going to result in blindness. Okay. So we know, right, given this, stimulate it, cut it out, right? We know that V1 is responsible for vision. And here, if we're looking sort of at this figure, we see that the eyes are forward, striate cortex is in the back, right? Where the occipital lobe would be. So we see that the vast majority of optic tract fibers terminate on neurons in the lateral geniculate nucleus, or LGN, of the thalamus. Okay, so that's here. Lateral geniculate nucleus of the thalamus is right here. The pretectum is responsible for reflex control of the pupil to light. Okay, so if like I shine a light at you, you're gonna have like a reflex, right? And it's the pretectum that's responsible for that protective behavior. Okay, and the superior colliculus, which is right here, is responsible for orienting movements of the head and eyes. Cool. So we're getting just a better idea of different parts of the visual system and what each part does and how it works with other parts. All right, we have different streams of information processing, the dorsal stream and the ventral stream. The dorsal stream processes information about the where of the visual stimulus. And the ventral stream processes information about the what of the visual stimulus. The dorsally located visual association neurons are responsible for spatial orientation, binocular fusion, depth perception, and the location, movement direction, and velocity of objects in space. So this is the where system or where stream. The ventrally located visual association neurons are responsible for recognizing objects and colors, reading text, and learning and remembering visual objects, such as words and their meanings. And this is the ventral stream, or the what stream. Here's another view of our beautiful brain. And this time I've just provided, again, just for reference, major Brodmann areas, okay? So we see the Brodmann areas for vision, audition, some of the sensory and motor cortex. And then down here, I've just provided more specific Brodmann areas for each of these more visually focused activities, right? So we see striate cortex is right here, BA17. And then the dorsal and ventral stream, right? The where and the what. We have the BAs for those. Okay, and then Extrastriate cortex is right here. And you can use this to help you identify all the areas. And I just like to provide you with different images, right? This is nice because it provides some of the terminology like posterior parietal lobe, whereas here you're just going to have more Brodmann areas identified for you. Okay, so go ahead and just get familiarized with these images. All right, we'll go ahead and discuss a little bit about the auditory system, and then we'll wrap it up for today. Okay, and we'll continue in the next lecture with the cognitive neuroscience of language. Okay, we'll be drawing upon some of the findings and points from today. All right, so this is our ear, right? An important part of our auditory system. This is the pinna. You can think of it as sort of like a catcher's mitt or a glove attached to the outside of our heads, sort of help collect auditory information or acoustic information, right? So the pinna collects waves of pressure changes in the air, slightly modifies them, right, given the structure of the ear, and directs that information, those pressure waves, towards the eardrum. The tympanic membrane is just another term for eardrum. So we see, right, we're collecting acoustic information, like maybe I'm playing you a song on the piano, right? From the piano, there's going to be waves of pressure changes going through the air, and it'll eventually go from my piano to hit your ear or to the pinna. It'll be collected by the pinna, funneled down towards your eardrum or tympanic membrane. Okay. The middle ear ossicles transmit movements of the eardrum or tympanic membrane to the fluid of the inner ear. Okay, so that's what the eardrum is going to do, right? It's going to take movements here, and then the middle ear ossicles are going to help transmit that towards the inner ear. The inner ear ossicles include the incus, malleus, and stapes. And we see that these here. Okay. And the inner ear also includes, importantly, the cochlea, right? A long coiled tube that contains the organ of Corti. And you can think about how these serve as nice amplification and transmission devices, right? From external stimuli into the inner ear, right? And then once it gets to the inner ear, there's still some work to be done. And we'll see that in the next slide. So the auditory system converts mechanical signals. Remember when I'm playing the piano for you, right? What's coming from the piano is pressure waves from the piano. The auditory system is going to convert those mechanical signals into electrical signals in the central nervous system, which is really cool. Hair cells, right? These guys, interestingly, have a characteristic frequency, which is the frequency to which the cell is maximally responsive. Notice that with the rods and cones, that they were also maximally responsive or tuned to certain things, right? So this is really cool that our perceptual system has components that are tuned in specific ways. And in the case of audition, it's hair cells with their characteristic frequency. Hair cells will usually respond to other frequencies besides their characteristic frequency, however, only at greater intensities, okay? So again, it's called the characteristic frequency because they're sort of selectively tuned to that frequency most, okay? So in response to that frequency, they have the greatest responsiveness. Hair cells, and I'll just talk about different hair cells and sort of what they do. Hair cells in the organ of Corti in the cochlea of the ear respond to sound. Hair cells in the cristae ampullaris in the semicircular ducts respond to angular acceleration or rotation of the head. And hair cells in the maculae of the saccule and the utricle respond to linear acceleration or to gravity, okay? All right, so we're talking about characteristic frequency here. You might ask, what is frequency? Well, frequency of a sound is associated with its pitch, okay? So if you hear different pitches in music, right, they're gonna be associated with different frequencies. And frequencies are measured in hertz or cycles per second. Physical characteristics of the basilar membrane cause different frequencies to reach maximum amplitudes at different positions. You can think of this as like a piano, okay? High frequency sounds stimulate the base of the cochlea, where low frequency sounds stimulate the apex of the cochlea. Tonotopic organization simply refers to the fact that cells responsive to different frequencies are found in different places at each level of the central auditory system, and that there is a standard relationship between position and frequency, just like a piano, okay? So when you lay out, right, the basilar membrane, there's going to be a particular part, right, that is in some sense like most resonant to a particular frequency. And just that layout, right, is called like a tonotopic organization, just like on a piano, right? At one end of the piano, it's very high pitched, and the other end, it's very low pitched. And on each place of the piano, there's a sort of relationship between position and frequency, okay? It's the same thing with the major components of our ear, which is really amazing, right? And you can see that through here, right? So this is the basilar membrane, and we see that each part is tuned in a particular way, right? So we see that there's 5,000 hertz here, 7,000 hertz here, 20,000 hertz here. And if we go towards the inside, right, towards the apex, we get like 200 hertz in the middle, okay? So we see sort of like we have a piano thing going on, but it's curled around, okay? And we'll see this a lot in the brain too. Like we got to make room for all the stuff in our brain, right? So you got to like tie things up and fold them together. Some cool origami going on in there. In general, all auditory afferents synapse in the cochlear nuclei and in the thalamus. And the auditory system has a fast-acting and slow-acting system. Okay, just like we had pointed out before that there's a nervous system and an endocrine system, and the nervous system is fast and the endocrine slower, right? And then we have different aspects of the visual system, right? We have that which is more responsive to color and objects, and that which is more responsive to motion, right? And low illumination. Likewise, here in audition, we have different systems serving different purposes or functions, okay? In the auditory system, we have a fast-acting system, which may function as a general warning. For example, as when you might jump from a loud sound, right? Like quick, okay? Fibers of the fast-acting system synapse in the dorsal cochlear nucleus. Whereas the slow-acting system may provide more detailed information about the sound, such as its location. And fibers of the slow-acting system synapse in the ventral cochlear nucleus. And here on the figure on the top, we have a map of the fast-acting system, and on the bottom, a map of the slow-acting system. And we just see that there's just a little bit more stops involved in the slow-acting system, okay? And although it may take longer to work through the slow-acting system, it helps us with identification of objects and whatnot auditorily. Okay, so cool. All right, we covered quite a bit today. And I just want to conclude with a reminder of some of the major things. Okay, so in the beginning of the lecture today, we covered neuroscience methods. We looked at lesion methods. We looked at structural MRI, functional MRI, EEG, and TMS methods. We looked at strengths and weaknesses of each method. Okay, we also considered major component parts of the brain. Okay, we also looked at the different lobes of the brain and the Brodmann areas, as well as their corresponding functions. Okay, we saw that the occipital lobe is responsible for vision. It's the home of primary visual cortex. We saw that the temporal lobe is largely responsible for audition. It's the home of primary auditory cortex. Okay, the parietal lobe is largely responsible for the somatotopic organization of our body, right? So you'll have a nice homunculus map here in the parietal lobe. Okay, the somatosensory cortex is located here in the parietal lobe, and the frontal lobe is the home of the motor cortex. The frontal lobe is responsible for our executive functions. Okay. We also notice that there's ways that we can separate the frontal lobe from the parietal lobe, right? And we use the central sulcus of Rolando for that. We can separate the parietal lobe from the occipital lobe with the parietal occipital sulcus, and then we can separate the temporal lobe from the frontal and parietal lobe using the Sylvian fissure. I also wanted you to sort of just appreciate Brodmann's areas, and for today at least, try to memorize these four, right? BA17 in occipital lobe is visual cortex, okay? BA41 in temporal lobe is auditory cortex. BA3 in parietal lobe is somatosensory cortex. And BA4 in the frontal lobe is the motor cortex. Okay. Awesome. Now that we have a very basic understanding of neuroscience and neuroscience methods, we'll continue our discussion in the next lecture by focusing on the cognitive neuroscience of language. As we continue our discussion of the cognitive neuroscience of language, we will revisit some of the main concepts from this lecture, including major areas of the brain responsible for visual and auditory processing of language. Although we can spend an entire semester or several semesters or a lifetime studying the neuroscience of perception, action, and emotion, our goal in this course is the more modest one of gaining an overview of the brain, its major components, and its different functions in order to understand higher cognitive functions, which is our main goal as cognitive scientists. Well, thank you so much for your attention today, and I hope you enjoyed the lecture today. I look forward to seeing you tomorrow for our next lecture.